Yeah.
Hello, and a warm welcome to everyone. My name is I am Farah. So we are before we get started, let's go and audio of our text will take the time. So the for your information, to avoid interruptions, everyone logging in is muted and the video is disabled by default. And when applicable, participants will be unmuted or the video will be enabled. Nonetheless, you may communicate via the chat box. Thank you for joining us for the Bioinformatics Workshop Fiesta, or BWF Colloquium, an inauguration event of the Fiesta. The Colloquium is co-located with the Malaysian Youth Bioinformatics Symposium 2.0, organized by Perdana University to University. For your information, BWF is a joint effort involving the Association for Medical and Bioinformatics Singapore, MYBS in short, Asia Pacific Bioinformatics Network, AP Bionet in short, and Perdana University, Malaysia. BWF aims to expand bioinformatics skills and competencies in the Asia Pacific community and beyond. MYBS is working towards same goal locally in the region. There are others acting the local efforts in the region and more needs to be done. The BWF series offers a plethora of 16 workshops of choice with focus on field. For more information, do visit the BWF website. The links are also provided in the Zoom chat box right at the top. Without further ado, we would like to proceed with the welcome address and call upon Dr. Muhammad Assoc, the President of AP Bionet, to give his address on behalf of the BWF Working Committee.
Thank you, Lisa and Farah. So, hello and a warm welcome to one and all. Thank you for taking the time on a Saturday. I know it's a big toll for many, so thank you for sparing the time on a Saturday to join us for this event. And representing the BWF Working Committee, comprising of Bionet, MYBS, Perdana University, there are three key messages that I would like to share. First, kudos to the organizing team for making this event happen. The team is largely made up of undergraduate and some postgraduate students. And for many of them, this is perhaps their first time organizing such an event of this scale. We strongly believe in providing students with opportunities for training and exposure. And we hope this was an enriching experience for all of them. Secondly, I want to give a quick history of BWF. The first BWF was successfully started and completed in 2018 with 9 back-to-back workshops, 1 per month. And this was actually in collaboration with our partner, Universitas YARSI in Indonesia. Back then, actually, we did it in a different way, different from today. It was the classical way, face-to-face interaction with the participants. And we still managed to attract more than 190 registrations. For all, that's the total for all the 9 workshops. We also had a colloquium back then, which was attended by about 60 plus participants. So we wanted to repeat this success in 2020 by bringing it to Singapore. And then, as you all know, COVID-19 happened, and that changed everything. However, instead of, on one hand deciding to cancel the plan, we decided to take up the challenge and wanted to go virtual and offer it to a much broader audience, which is something we have always wanted to do, but this felt like the perfect time to do so. Of course, and today is the outcome of that effort, and this outcome is both exhilarating and intimidating at the same time. So this map you see here speaks volume. We have close to 700 registrations for the colloquium alone, which is way beyond the earlier number of 60. So this is really heartwarming to see that many people are interested from different parts of the world. You can see the numbers there. So we thank all of you who have taken the time to do this. And for some of you, this may be a little odd hour. Some of you may still be very early in the morning. So it is exhilarating because this was a dream. It was a dream to reach out to such a large number at a single event. And at the same time, it's intimidating because this is our first, and of course, we are worried that things will go wrong here and there. And so I'm going to apologize in advance for any shortcomings on our side during the course of the evening today. The third point I want to highlight is that BWF is a joint effort, and it aims to grow bioinformatics skills and competencies in the community. For a series of 16 workshops, we have a path where we actually break down the workshops into essential, beginner, intermediate, and advanced, so people can start off wherever they feel more comfortable, and those who are really new or just starting, they can start right at the beginning with the essential and go all the way to the advanced workshops. So we have also, for the instructors of the workshops, we actually have advertised across the region, so we aim to get the best instructors to be facilitating workshops. We got a very good response. We are working with them to finalize the schedule for the workshops, the schedule very soon. And we hope many of you who are participating today would actually partake in the workshops eventually, the first one starting next month. So do look out for our future announcements. With that, I thank everyone for joining today. I hope you have a blast and have fun and learn a lot from the session today. I thank everyone involved in making this happen, and not forgetting our workshop instructors, our keynote speakers, and the plenary speaker who also have taken time to speak to you today and share their wealth of knowledge. Thank you very much. Zita and Farah, I pass it back to you.
Thank you, Dr. Asif. Next, we call upon Dr. Yang Waikid, Acting Dean of Perdana University, School of Data Science, representing the Perdana University, as the main host of MYBS 2.0 to deliver a welcome address.
Hi, good morning, everybody. I hope you can hear me. If you could, maybe just give me a thumbs up. Cool. Thank you so much. So, hi, welcome to this event. So, on behalf of the university, we would like to thank all of you to be here. Again, like what mentioned by Prof. Asif, that it's a bit unique in the sense that we are doing it virtually. But I'll see this as a new normal, and I think that it's a new way for all of us to embark in this journey. So, before that, I want to thank the organizing committee for this event for able to pull everything in a very, very short time. And just mentioned by Prof. Asif that this is very unique in the sense that it was initially planned to be a physical event, but because of the current condition, it has changed into a virtual condition. So a lot of things has changed, but I'm so happy that the team is very versatile and they are ready to move and make the event to be a success. So thank you very much for the team. And I think I'm not going to talk more about BWF because Prof. Asif already mentioned, but I'm happy to share from the Perdana University point of view that this BWF Bioinformatics Workshop Fiesta has been a project. It's like our baby for our university, and with just one aim to be able to go around to train people with various fields in bioinformatics, and we have done it successfully in 2018. 2019 as well. So this year, initially the plan was to do it in Singapore physically. However, because of the current situation, but I do not think that. I think this is great in the sense that it becomes virtual and we can involve many other people around the world. So, well, we take opportunity of current situation, and I feel that this is the right way that we are moving forward. So, good job to the team. So, just let me talk about bioinformatics. After all, we are here for a reason. Bioinformatics is such an exciting field, yeah, that it focuses on interdisciplinary field. So if you check on your friends who are in another field, they probably very much focus into a specific topic. But bioinformatics students are trained in various fields, ranging from biology to computer science, mathematics and statistics, and even many more. So you can see that we are actually at two different, very, very wide spectrum of the field that we have. And this has become students to be very, very versatile. Yeah, so I always mention to students that being versatile is important in the current setup because we are no longer moving into a silo, but we are all moving into an interdisciplinary kind of world. So it's so important that we are trained in different perspectives. And if you see the current trend, not only that bioinformatics is so important, but how big data has come into this perspective. And in this current world where we're in this big data and also data science, we can see that bioinformatics are like forerunners because they are trained in looking into data and not only look into data but also transform them into informative information. So, and this kind of technique is so important currently in this current setup. And therefore, because of that, I think as a graduate in bioinformatics, we should be proud of ourselves that we are doing this. We are actually at the forefront, and this is the part that we come in. But however, coming from a university point of view, I strongly believe that it's so important that you have a continuous learning attitude. That is why the university's continuous effort is that we wanted to train people in this field. Having just a graduate, having just a bachelor degree in bioinformatics perhaps is insufficient. From time to time, we need to learn, we need to upskill ourselves. Therefore, our university, we are always very active in providing the opportunity, not only for students, but also for researchers or even for those who are working at various stages of your career, that we train you at different levels. So, like for example, in Perdana University itself, we have formal programs ranging from undergraduate to postgraduate. And that commitment of doing that education is supported by even giving scholarships, discounts, and also financial aids who wanted to be in that field. So those are formal programs, but we also have a lot of informal programs where we allow you to have training from time to time to upskill yourself. Therefore, it's so important, to be in this network and to learn from time to time. So we actually encourage those active participation, yeah, between associations and societies. Therefore, the connection is important and networking is important. Having an event like today, I think this is a very good thing that we are all connected and we can learn so much from each other. So with that, I want to thank all of you, and again, I want to wish you all the best, and I wish that you have a good time today in today's event. Thank you very much.
Thank you, Dr. Yam. Last but not least, we welcome Ms. Shalini to say a few words as a voice of the student community.
Thank you, Ms. Farah and Ms. Sita. A big welcome to all of the participants, speakers, and instructors. Thank you for joining us today. I am Shalini Kumar, the chair of the MYBS 2.0 organizing committee. I thank the opportunity to say a few words on behalf of the community. Events such as these are vital to the student community, which provide a unique opportunity for all of us to meet people from our field of interest, to share knowledge and experiences. Indeed, there is nothing more exciting than meeting new people, hearing their stories, and being inspired. In the current COVID-19 pandemic, this has become more difficult, but we are grateful of the virtual platforms that allow us to continue to engage with others, albeit a little differently. Nonetheless, the challenges of this new norm bring along new opportunities. For example, 8 months ago, I did not imagine that I will get to attend a bioinformatics event together with 600 plus participants from different parts of the world. Come to think of it, it does feel surreal, at least to many of us students who are more used to the classical way of teaching. I would like to take this opportunity as well to say a few words as a student of Multimedia University. We students of the Faculty of Information Science and Technology in Multimedia University had the honor to organize the first MYBS in November last year. We didn't expect there will be another MYBS this year because of the pandemic. We would like to thank Perdana University for inviting us to co-organize MYBS 2.0. We hope that another university will consider to continue this with us in the future. And I think the future looks bright for bioinformatics and data science students with the rising demand for more people with the skills and competencies to manage and mine data. I am pleased to have MYBS tie up with BWF in support of the systematic training program that has been planned ahead for one whole year starting next month. As for today, we have a great live program, and I hope it will be fun and full of learning for all of you. And I made to understand that we may have some surprise announcements. Please do look out for them. Have fun and all the best. Thank you. So let's pass back to Farah and Zita.
Thank you, Shalini. We also want to acknowledge the effort and contributions of our partners, National Supercomputing Centre Singapore and Alta Coast Sdn Bhd. We thank both organizations. With that, we conclude the welcome ceremony. Declare the event formally open and hope that you enjoy and benefit from the program plan. Our first bioinformatics activity for the day will be 2 workshops that will be run in parallel. The first workshop is entitled Biomolecular Visualisation, which will be delivered by Dr. Choi. Dr. Choi is an associate professor at Perdana University and the chief executive officer of Alta Coast Sdn Bhd, a local bioinformatics startup. With a degree in biochemistry from University of Malaya, she pursued further studies with a master in information technology and PhD in pharmaceutical technology from University of Science Malaysia. Besides varied research experience, she was the facilitator for international students from University of California, San Diego, United States, Osaka University, Japan, Indian Institute of Technology, Delhi, India, Chulalongkorn University, Thailand, and many more. and many more prestigious universities around the world under the Pacific Rim Application and Grid Middleware Assembly Program. Prior to her appointment at Perdana University, she was a research scientist at the Malaysian Institute of Pharmaceuticals and Nutraceuticals (IPAM) and a postdoctoral research fellow at the School of Industrial Technology, USM. Her expertise include protein structure prediction, membrane molecular dynamic simulation, and in silico high throughput screening, as well as structure and ligand-based drug discovery. Of late, she has been making a mark in other areas of bioinformatics.
The second workshop is entitled Healthcare Data Analytics, which will be delivered by Dr. Kenneth Van Horn Kim. Dr. Kenneth obtained his Bachelor in Biochemistry and MYBS degree from the National University of Singapore and a PhD in Cancer Biology from Stanford University. He completed his postdoctoral training in mouse carrier genetics and at the Institute of Molecular and Cell Biology, IMCB, Singapore, before joining the Department of Biochemistry at NUS 2011. He also serves as the Program Director for Life and Biomedical Science at the National Supercomputing Center, NSCC. His research and education interests are in cancer genetics, high-performance computing, for precision medicine and bioinformatics education. He is currently the elected president of the Association for Medical and Bioinformatics Singapore, MYBS. That was a brief biosketch from the illustration career of the true instructors. Some housekeeping announcement before we begin the workshops. All relevant links for the program today, including the workshops, are available in the welcome letter, which was emailed to you. If you did not get them, and you are here because someone shared the link with you, you may want to check your spam mailbox because it has the other key information for today. By the way, you can access the Open Broadcast YouTube channel link from our BWF and MYBS websites, or the links provided in the Zoom chat box. Please use the chat box sparingly, only for questions and if you need any help. Also, we kindly request again that you change your Zoom profile name to your full name so we can address your actual name during the Q&A session. With that, we now hand over to the instructors and we'll be back for some housekeeping message at the end of the workshop sessions. Have fun all.
Right, thanks very much. Thanks for the introduction. Very happy to be part of this inaugural BWF 2020 session. This, as Professor Asif Khan mentioned, this is something new, the new normal for us.
Hi, good morning. Good morning, everyone.
Morning, but they can't unmute themselves, so maybe you can start the workshop one now.
Okay, great. Okay, let me see this morning how many of us we have here. We have about 50 of us here. Okay, good morning to everyone. First of all, I would like to thank the Miss MC, both Zita and Farah, for a kind introductions. I'm Sibing. Very good morning to all of you here who join us here, and I would like to thank the organizing committee for giving me such a great opportunities to share what I have acquired during my research journey. Thank you for BWF organizer, working day and nights for the past few days to solve all the technical problems. I am actually overwhelmed with the number of participants that have registered, to be frank. So this is such a big crowd, I would say. We have two parallel sessions, Dr. Kenneth, the other side, and me, myself here. How many of us? We have 50 now. Okay, so let me share my screen to kickstart the workshop. Just give me a few seconds. So can everyone see my screen here? Yeah. Okay, good. Let me blow it up. Okay. So yeah. Okay. So everything is cleared. Okay. So let's start our workshop because we are running a bit back from the time. Okay. So now, yeah. So before we kickstart the workshops, I would like to do a brief introductions on the institute that which I'm actually affiliated with. From the introduction, probably you have heard, I'm from Perdana University, attached with School of Data Science, School of Data Sciences. So we are Perdana University is relatively a new university. We are only 8 years old, and for School of Data Sciences we are only 5 years old because we kickstart as a Center of Bioinformatics from the beginning. So we evolve, we grow, and then in 2015, School of Data Sciences has the Center of Bioinformatics sort of like grow up and matured and become a School of Data Sciences. So in terms of educational pathway, we have a new baby which have we have delivered. I would say born last end of the year. We have this formal educational program, bachelor degree in computer science, majoring in data science. Okay, this is a 3-year program. And with this program, we have we do have internship programs. So for those who are interested to join us for their internship, do welcome to write in to us. Okay, and the next program is we have postgraduate diploma in bioinformatics. This is a 1-year program, postgraduate programs. And from there, we have 2 of our master program, master in bioinformatics by research, as well as master in bioinformatics by coursework. So both of them is ranging 1.5 years to 2 years program. Okay, so then from there, if those who are not keen in going into a formal educational program, probably you can join us for our short courses. We have the DSX bootcamp, workshops. Okay, from time to time we have free talks. Okay, so all these short courses, workshops, we run it throughout the years. Okay, from 2 months, 1 to 5 days. Okay, so do write in, inquire about all these short courses. And last but not least, the ultimate aim, we do have our PhD program, which runs for 3 years. Okay, so for more details, do visit our website with the URL written at the bottoms of the slides. Okay, so so that is about our educational pathway in Perdana University School of Data Sciences. So before I kickstart, I have a good news to share with everyone. Is currently our university is running a 50% scholarship for all postgraduate programs. So for those who are interested in pursuing their study, to further their studies into postgraduate diploma, master in bioinformatics by research, by coursework, do write in and write in to inquire about our program. So for me, frankly, this is a very good opportunities for those who are interested and keen to do the postgraduate studies. Okay, so I think that's enough. I hope everyone don't get bored, and then we are going to start our workshop now. Okay, so let's move on. Okay, so I guess everyone must have been heard all these phrases. Okay, show, don't tell, or a picture is worth 1,000 words. Follow on with the soul never thinks without a picture. A good sketch is better than a long speech. And for those who have who know Mandarin or Chinese, probably you will understand what is the last phrase meant. 百闻不如一见. Okay, all these phrases is telling us that visualization is better than what we can visualization is a great way for us to imagine. It's a great way for us to understand more in depth of certain stories, understand more in depth in terms of speech if you talk, or understand more in depth if you read as compared to if you read a book. Okay, so from there you will know that what is the reason behind that we are doing a visualization of biomolecules. Okay, so move on. We will have so let me show you something here. If you have a picture like this, how much you can tell from this picture? Probably you will tell me a bunch of powder. Okay, from this image, probably you can even further tell me that, oh, this is a coarse kind of powder, or very fine, very smooth powder, and in terms of the colors, it is white. Okay, but what if I'm giving you something like this? Okay, how much you can understand with this bunch of text as well as this bunch of numbers? Okay, for those who probably have some what you call some biological backgrounds, you probably can know from there you will know, oh, this is a 3-letter representations of amino acid. Probably is leucine, isoleucine, histidine, tyrosine. Probably for those who have some knowledge in terms of biology, you will make a guess, well, this is some kind of protein, I suppose. But how about all these numbers? How much you can tell me with all these number? And for those who have some chemistry backgrounds, maybe you can make a guess. Well, the C probably is a carbon, the N is a nitrogen, and O is oxygen, right? So, but what does all these numbers tell you? Okay, so probably you wouldn't know what is that all about. Okay, without further transformation of this kind of data. Okay, so if this data is being transformed into an image like this, how much you can tell me? Probably you can explain to me, well, I'm not sure what is that. Is it a bunch of noodle, I guess? Or for those who have some also some knowledge from text, biochemistry knowledge, you probably make a guess, well, this is a protein which is in a secondary structure representations, and some part of it is a helix, some part of it is maybe this is a beta sheet. Okay, and this probably is a kind of chemical compound or ligands. Yeah, so you make a guess. Well, from that piece of data earlier on that I show you with all the text and also number, this is being transformed into something where you can explain more on it if you have some knowledge on it, right? Okay, what if I'm giving you this? Okay, with the current technology, okay, this is printed out from a 3D printing. Okay, so this is a 3D printing of a dengue virus, if I'm not wrong. Okay, and this is being printed out in a 3-dimensional way where you can hold, you can feel. Okay, so this will explain more to you in terms of the additional dimension where, or probably I will know that the spike protein, where is it located? Okay, where's the capsid? Okay, what's the shape of this particular virus? Okay, so you will have additional informations if you have this kind of visualization aids or tools, okay, for you to feel and for you to visualize. Okay, so furthermore, we have augmented realities nowadays with some kind of 3D goggles. You can even see the interactions of one particular proteins where it is interact with some kind of ligands, whereas the binding site, you can even imagine yourself with the augmented reality going into the binding site, look at the cavity of the binding site, identify what are those interacting residues that involve in the binding site. All those can be done with the current technology. And what is the reason behind all this technology? Okay, this is because visualizations is a kind is a tools for us to communicate. With the visualizations tools that we have, we can see 3D structure, we can communicate features about the structures, we can illustrate the biological process. For example, as what I mentioned just now, you can see the catalytic, you can identify the catalytic site, you can understand how the bindings actually works. Okay, with also the 3D printing, augmented reality, a layman can actually feel, oh, this is a protein, how the protein is being visualized with the goggles, with the augmented realities. Okay, so that is the reason that we are doing visualizations. In particularly in this workshop, we are focusing in biomolecules. Okay. So the overview of these workshops, okay. So the main objective of this workshop is I'm trying to share some user-friendly software where is totally free and publicly available, where everyone can use this to produce or to understand how the protein interact and to visualize the shape, the what kind of protein is that. So all this software that I am actually introduced in this workshop is actually freely available. It's an open source software. So we are going to look into VMD, Pymol, Ligplot, okay, and from there you will pick up how to create a public how to pick up how to create a publication quality molecular image. So you will learn how to render an image, okay, and the level and pace of these workshops, okay. So this is designed for almost everyone, from faculties, postdoc, research staff, students, okay, with some basic knowledge in biochemistry, and but with no previous experience in molecular visualization software. Okay, so this is designed to cater for those who have no experience in molecular visualizations. Okay, so at the end of the training today, I hope that all participants should be able to use a free visualization tools to generate images in the in your work as well as in your research. And you should be able to create different representations by manipulating selections with protein structures. And at the end of the day, you should be able to render high quality graphics. You should be able to render interactions diagrams. Okay, this at the end of the that is what I am aiming to achieve at the end of the trainings. Okay, so everyone clear about that? Good. Okay, so I guess everyone have received email from the organizing committees that some instructions before attending the workshops, where we are actually expecting all participants to have all three software, all three tools installed in your local PC or laptops. Okay, so the instruction is given with the hands-on materials, I guess. 3 materials I'm distributing to all of you out there, where the installation guides as well as the tutorial for all 3 different tools. Okay, so I guess I'm assuming that all of you have the software ready in your laptop. Okay, so Before I go on with the demonstrations on how we so-called go on with the tutorial, I will do a general introduction of all three tools that we'll be using in the next couple of minutes. Okay, so the very first tools that I'm introducing here is called Visual Molecular Dynamics. Okay, this is developed by University Illinois, Urbana-Champaign. Okay, so this visualization tools is freely available. You can register. It's free, but you need to register. Okay, so with the instruction given to you in the handout, okay, I suppose if you have it, okay, later during the demonstrations, I will walk all of you through the tutorial or the hands-on materials that I distributed earlier on. Okay, so the first part of the tutorial is actually the installation guide. Okay, so I hope everyone managed to download the software. Okay, so what this software can do for us is, okay, it helps us to visualize and render the molecules, and it also set up and write out the molecular dynamics input file. This is if you have integrations with VMD together with NAMD. Okay, so this software is developed if you are using, I should rephrase my statement here. Okay, this software can analyze or can run together with NAMD if you intend to actually generate the input file of NAMD, okay, to as well to analyze the output of NAMD. Okay, so this is also a free software. We have active development or user community where if you have any issues on installing the software, you can always go to their mailing list and go through the list of communications that have been done by other users, that what are those problems they have faced, probably from the response from the developer. You'll know how to troubleshoot your problems while using VMD. Okay, so it can use in different platforms. Okay, so no problem with Macs. Okay, I'm using Mac. I have a Windows machine as well. VMD works fine. Okay, so if Mac no problem. Linux, Mac is developed using Unix platform. Linux have no issue also, so it's a cross-platform viewer. Okay, so and the best thing I love about VMD is where it's not so heavy. Okay, I would say in terms of the processing, the computational processing, VMD, it won't give us so much problem in terms of maybe you run halfway of the program, it hangs. Okay, this VMD seldom happens unless you are visualizing a huge or big number of trajectory. Probably this will give you some problems if you don't have a powerful laptop. I would say the memory is not enough, then this will actually give you some problems. Okay, but in general, processing like loading a single PDB files, okay, doing some analysis on the PDB files, put in position and all this, VMD works perfectly fine. So I actually learned visualizations. This is the first software that I've learned during my training, PhD trainings. Okay, so I actually love VMD so much. But to be frank, if you want to compare VMD with some other commercialized commercial software, it is not so user friendly, okay, as compared to the commercial software where you need to pay. But somehow, whatever the commercial software can be done, VMD is doable as well. Of course, additional steps needed. Some cleaning of the data is needed. Not as user-friendly as compared to the commercial software. Okay, so this is about VMD. Then the next one I'm going to introduce is Pymol. Pymol is also where Pymol is the tools that I have picked up recently. Okay, all this while I have been using VMD, and to be frank, Pymol generates very nice images for those who likes to have nice images in your publications, in your poster presentation, in your slides. Okay, I guess Pymol can do a very good job on this. Pymol is one of the first batch of the tools that develop on top of Pythons. Okay, and it is also allow user to actually put in some plugins. If you know Python's scriptings, you can even write the code, okay, or the Python script, which can help you to do your analysis, where this thing can integrate into Pymol. Okay, and Pymol also support one of the rendering techniques, which is called what you call ray tracer. Okay, so this is a rendering techniques that help to mimic the lighting when you generate the images. So Pymol do a great job on this ray tracer. So that is why at the end of the day, Pymol can produce a very high quality images when you have different layers of, for example, a surface on top of ribbons. So all this can Pymol can handle it very nicely. And the best thing is this is also freely available for academics, for educators, as well as for students. You need to register, okay, but somehow this is free. If not, if you are not a student, probably you will have only a trial versions, which is maybe a 30 days trial. I cannot remember the durations that Pymol allowed, but still, I think the 30 days is sufficient for you to explore and to understand, or to get a rough idea how powerful Pymol is. Okay, so this will be the two visualizer that I will be introducing to you later on. And the third tools is Ligplot Plus. Okay, this is a tools that allow us to analyze the interactions between a protein and also a ligand, or a protein with the substrates, or a protein with proteins. Okay, but in this workshop, I'm only going to go into a protein and a ligand interactions. So what this program do for you is they predict, or they analyze the interactions between your protein and your ligands, and produces a 2D interactions diagram, which looks like what you have here. Okay, so this is a Ligplot program, and yeah, this is the three tools that I'm going to introduce to you. Okay, so now I'm going to explain a little bit on the subject that I'm going to use in all our three tools that you will be learning in a while. So I'm going to use a protein which is called hemagglutinins, and this protein is actually an influenza virus protein. Okay, it's called and the PDB ID is 1RUZ. Okay, and probably someone might not familiar with PDB. Okay, so let me add up a little bit on PDB. Okay, Protein Data Bank. PDB stands for Protein Data Bank. This is a database that actually stores all the protein structure which is being elucidated either via X-ray crystallizations or NMR techniques. So the structure is being elucidated, the crystal structure, the X-ray crystal structure, and then the NMR structure is being elucidated, and all this structure will be deposited into a protein database called Protein Data Bank. Okay, so we can obtain the protein from this protein data bank. Okay, so and throughout our workshop this morning, I will only utilize this protein, and we will go through, we'll use this protein to generate images using VMD, using Pymol, and also by using Ligplot, you will look into what is the ligand that interact with the hemagglutinins. Okay, so far so good, everyone. Okay, I hope everyone can answer. No, can understand so far. Let me check whether I have any. Okay, so all good. Everyone is good. So let's move on to the demonstrations. Okay, so I'm going to share my desktop. The reason I'm sharing I'm sharing the desktop is because some of the program that actually pop up several windows, so I can't be sharing one window by window. So I'm going to share my entire desktop with everyone, and I'm going to start off the workshop by doing the demonstrations on VMD using the hemagglutinin protein. Okay, so before we start on that, I guess probably I think I will proceed. Okay, the break will only be after the VMD sessions finish. Okay, so I will be giving out a 5 minutes break. So let's unshare this, and I will share my desktop. Okay, good. Okay, so I'm sharing my desktop now. Everyone can see my desktop? I need to get some response from my moderator. Okay, all good? Can you see?
Yes, yes,
Okay, good.
yes.
Thank you. Yeah. So now I'm going to launch my VM. Okay, but before that, I would like to introduce the protein data bank with you all. Okay, so let's go into a protein data bank. This is the URL for protein data bank. Yeah, this is the one, www.rcsb.org. Okay, this is a protein data. This is how a protein data bank looks like. Okay, and inside this database we have over 100 over 1,000 if I'm not wrong, 150 over thousands of crystal as well as NMR structure of the protein. Okay, so you can do a searching by typing the keywords here. Okay, or if you know the PDB ID, like what we are doing in our workshops, we are using 1RUZ. So this is 1RUZ. So what you need to do is just typing the ID into the search panel and hit the enter. Okay, so you will see 1RUZ here. Okay, so they are telling you this is 18 H1 hemagglutinins. Okay, so what we need to do here is we will need to go into this protein, okay, and download the 1RUZ PDB file. Okay. Okay, so you'll be seeing my screens where this is all about 1RUZ. So if you browse through the web page, this is the structure that the publication of these structures is here in the literature panel. It's being published in 2004 by Skihel JJ, okay, and co-workers. And the publications is here. And if you scroll down some more, you can see the information about these protein structures. Okay, this molecule is with the name hemagglutinin, and it consists of H, J, and L with the sequence length of 328 residues. Okay, and this belongs to influenza A virus, and what's the location that is being found is in South Carolina. Okay, so there's a lot of information about these structures. Okay, if you scroll down some more, all this is the entity ID, okay, and all the protein features, the sequence, okay. And if you scroll down some more, you will see that small molecules actually mean what are those ligands beside the protein? What are the ligands that bounded with the protein? Okay, with the hemagglutinins. So in these structures, we have two ligands, okay, that bounded with our protein. One is with the identifier of NDG, the other one is NAG. Okay, so NDG is 2-acetamidol, 2-deoxy alpha D glucopyranose. Okay, so these are the ligands that actually bounded with our hemagglutinin. And from here, experimental data, you will see that, okay, this is a X-ray diffraction structures. What is the resolution? The resolution is 2.9. This is not a very good quality structure, I would say. So if you look at your right-hand side, okay, this colored bar, okay, this is actually a structure validation in terms of the secondary structure arrangement of this model. Most of the score is actually fall below below average, which is in the red region, and this is also indicated that this is not a very good structures. Okay, for those who understand about Ramachandran's analysis, Ramachandran analysis is an analysis where we actually look into the backbones arrangement of the proteins. Okay, so when it's telling us the outliers is fall into a red region, it actually means that the secondary structure or the steric clashes of this molecule is not that good. Okay, so maybe the structure is not in favorable conditions. It's not no, I should say the structure is not in the optimal conditions. That's why the scoring of the backbone arrangement is not that good. Okay, so when the data is being deposited, this is the date. Okay, and who are those authors that deposited? Okay, so revision history is all here. Okay, this is about the database, and this is about 1RUZ structures. So what we need to do is we need to go to the downloads and download the PDB file. Okay, this file will be used in our VMD visualizations. So I hope, so I'm going to download this file. So what you need to do is you will need to click on the PDB format, and then the 1RUZ is being downloaded. Okay, so we will be using this 1RUZ later. Okay, so this is the data that we'll be using in our next 2 hours. Okay, so we are done with PDB. Now let's move on to VMD. Okay, so far so good. Okay, let's move on. What should I do now? Okay, so what we will need to do here is I will need to share open up my tutorial. Let's see, where's my tutorial? Okay, so this will be my tutorial. I'm going to put it aside. I don't think I can do that. Okay, so then I'm going to launch my VMD. Okay, so my VMD is being kept in applications. So I'm going to launch my VMD here. for those who are using Windows, I guess window will be much easier. Okay, just double click the VMD icons, then you will get your your VMD executed. Okay, so the first part is installation. If you have your hands-on material with you, okay. So I'm walking through the tutorial with all of you. So what you have here is the first part. Okay, I'm giving you is the installations. So the installations, I'm assuming everyone have done it. Okay, and all the program is running perfectly fine. Okay, so now I have launched my VMD here. So when you double click the VMD icon, you will actually see three windows being what you call being executed. Okay, so you have a main GUI panel, okay, which is this VMD main. Okay, you have a so-called VMD console, okay, where this will be all the scriptings. If you have scriptings, you can utilize this windows, and you will have an OpenGL display, which is here. Okay, so this is the three main things that you will see when you launch your VMD. Okay, so what will have, so the main control will happens in VMD mains. Everything, file, molecule, graphic, display, when you click one by one, you will look at what are those functions that will have in VMD. Okay, so let's walk you through with the with this VMD main. Okay, so most of the time we will use this main VMD main to control what we wanted to do. Okay, for example, when we want to load our molecule in, okay, we will be using this. Okay, so I'm not going to go through line by line with the hands-on. Maybe after this workshop, all of you will need to spend some time on going through the hands-on line by line. But here, the demonstrations that I'm going to show you is a basic kind of operations, basic kind of usage in VMD, where how you should load your file. So I'm going to show you now what you can do in changing the colors, changing the representation, how you are going to render the images. Okay, so let's kickstart off this thing. Okay, so now let's go to VMD main. In order for us to load our files, okay, so we will need to go into file, just like what you have been doing in your Word, in some other tools. Okay, go into new molecules. Okay, browse to 1RUZ, the one that we have just downloaded. Okay, for me, do I have the 1RUZ here ready for me? If not, then I will need to go into my download. Because I have just downloaded 1RUZ. Okay, so so once you have typed in your 1RUZ, okay, browse the file, type and click load, and then 1RUZ will be loaded up automatically. Okay, but somehow, well, I guess everyone must be wondering, this is not the kind of images that we have seen in journals, in textbook. Yeah, this is not, because this doesn't mean anything. You will see only a bunch of lines that doesn't actually tells you anything. Okay, so what we'll do here is I will be showing you how you can actually change the representations of the molecules. Okay, so we will be going through page number 5. Okay, so in your handout material, page number 5, how we load the molecules. Okay, so what I have just done just now is I'm going to File, Molecules, browse, and then browse to the molecule that you have kept your 1RUZ, and then click open. Then you will see your file being loaded, okay, into the OpenGL display, which is here. Okay, so from there, okay, let me introduce some of the functions that you can play around in your main Windows. Okay, so I guess most of you are using your mouse, right? Okay, so if you go into the mouse, okay, you want to rotate the entire molecule, you can just go to mouse, rotate, then using the mouse, you'll be rotating the entire molecules. Okay, so if you want to do something like translations to move our structure left and right, up and down without moving the rotational mode, you can actually go into mouse and click to translations mode, then you will have translations. Okay, and if you go to mouse scale mode, then you will be actually enlarging and shrinking the structure. Okay, this is about how you can actually play around with the mouse control. Okay, so of course there are some others like center, queries, label. If you want to label one particular atoms, so you can go into labels, label atoms, bond. Okay, so let's go through the manual. Okay. Okay, so now the next steps that we are going to do here is changing the appearance, okay, of the molecules. So changing the appearance will be in here, this tab. Okay, just now we have used file by loading in new molecules. Here, this is by changing the representations. We'll be utilizing this tab. Okay, go to graphic representations. So you will see that this is exactly the windows that are in your handout. Okay, so this is how your structure is being represented in lines at the moment. Okay, so as I said, this is not a usual representation when you read through a journal or textbook. So what we can do here is we will change the drawing methods into something called new cartoons. Okay, ta-da, then you will have a structure like what you have seen normally, but we are yet there. We are not there yet. Okay, so what we will be playing around here is okay, so let's move back to this. Okay, so this, how we are changing the drawing style is where we look into this tab in graphic panels. Okay, so here is where we can change the coloring methods. For example, if you want it to be secondary structure, okay, so you can choose a secondary structure coloring methods. Okay, so it will change into the yellow, cyan, and also the magenta representations. Okay, so this will be, if you don't like a secondary structure representation, you can always change the color, like color ID, so you can play around with different colors. If you like it to be silver, then it become like this. If you like it to be pink, and then it will be like pink color. And you can play around with materials. For example, if you want it to be the surface kind of representation, then we will go into we'll go into what you call this is I will put it to opaque. That is a drawing method. So if you don't like it to be a secondary structure, you like it to be quick serve, then it will become like this. And probably you wanted to be something more advanced, like you wanted 2 layers kind of representation, then you can actually go into generating another representations, create representations. So now actually we have 2 representations here, and 2 of the representation is actually the same because we haven't changed the other yet. So you can either change here the first one, the second one, into a new cartoons, okay? And our quick serve, why is it you couldn't see the second representation? This is because our quick serve is yet to be transparent, okay? This is opaque, so now we need to change this into transparent. Then you will see something like transparent here, where the other layers of the cartoons is actually presented where you can see it, but because they are in the same colors, so we can actually change the colors into maybe a secondary structure representation. Then you will see something like that being projected. Okay, so let me blow this thing up a little, then you can see it clearly. Okay, then moving on, what we can do. Okay, so we can actually change the background. Okay, so if you want to change the background, then because of course black is always nice to have, because it will give us a very contrast kind of feeling, easier to identify. But somehow this is not the background that preferred by publications. So usually we prefer something which is in a white background. So what we can do here is we can go into the second tab, okay, which is the display. Okay, so we will go into we will go into graphics. Okay, not display, we'll go into graphics and colors. Okay, we'll go into colors, display, and go into background, and then you can actually change the background into white. Okay, so you will see your image being produced, transformed into like this. Okay, so now and I'm sure I'm sure the axis is not the thing that you prefer to sit along in your image. So you can actually turn off the axis here by going into display axis and turn it off. So the axis is not there anymore. And I'm not sure whether you have noticed, you are aware, the display is actually having some shadow kind. So I don't want it to be a deep depth queuing. So I turn that thing off by going into display depth queuing. And you can also change the orthographic or perspective, depends on your preference, by going into a display setting. Okay, so what else? Let me go through what else we can do with this. Okay, so now let's move forwards. Okay, so in graphic representation, go back to graphic representations. The drawing methods, actually there is quite a numbers of drawing methods that you can play around. Okay, in this demonstration, I have actually shown you like the what you call quick serve. Okay, as you can see, we are using quick serve. Okay, and we are using new cartoons as well. Okay, so there's 2 representation that we have play around, and you can also play around with cartoons. Okay, this is how cartoons looks like. Okay, some authors actually use these representations, and There are many, okay? So for those who are interested, you can actually play around with it. Yeah, so basically, this is how we actually manual the control panels, and there are some shortcuts also here that I would like to show you all. Is where, for example, if you double click, okay, on quick serve, okay, it actually turn into red, means that we actually hide the representations. Okay, by double clicking it, it actually show up again. So if I only want a quick serve, I don't want the cartoons, then you can actually hide your cartoons just like that. Okay, and why is it is like a blur kind of quick serve? It's because we are actually putting this in, the material is actually a transparent. Okay, the reason why it's being transparent is because we are actually overlaying two different representation on top of each other. So they are, if both of them is in opaque, you cannot see the one that are lying at the bottoms. So that is the reason that if we put it back into a quick serve, then you wouldn't see your cartoons, even if you unhide your cartoons. Okay, so that is how and where else? Okay, you can actually go into also here, where if you go into cartoons, beta sheets thickness. Okay, so you can play around with the thickness. If you want to change the thickness, and it will become thicker. The helix coil radius, then it will become bigger. Okay, and these are the things that you can play around. Okay, so far so good. Okay, so in my handout, you can see that I actually give some examples on what do you mean by line representations, van der Waals representations, Lycorize, new ribbons, cartoons, and new cartoons. So here in my demonstrations, okay, I can show you the representations. If you want it to be a line, it will become like this. If you want it to be van der Waals, it become like this. Okay, and likewise will become like this. So do explore and find the one that you think it will tell you the most story. Okay, that is the whole ideas of doing visualizations. We want to visualize in order for us to explain more, to understand more. So choose a representations where it will tell you the most. Okay, so what I usually like to do is I like new cartoons in secondary structure representation, because from here it can actually tell us the secondary structure arrangement of the entire structure. Okay, so there's some of the things that I think is quite useful is where here this probably some of you might ask, for example, if I want to select everything, okay, these representations, like for example just now when we load in the molecules, okay, we are using a representation of lines, and this is not, and is actually like this. Okay, so means that when we first loaded our PDB structure, the default representation is actually in lines, and is actually the selected atom is actually all. Means that if the structure, the crystal structure consists of water, consists of protein, consists of ligands, then you will have everything in your display windows. Okay, so in order for us to select atoms, you can actually go into these panels. Okay, for example, if you want to select only proteins, okay, this is the keywords for proteins. Then when you type proteins, then we will have all the water molecules disappeared, as well as some other ligands, okay, disappeared. It only display the windows. Okay, So that is the keywords. Let's say, for example, if you want to select some particular residues, for example, res ID, the first residue, res ID number 20. Okay, so if you only project the residue, okay, let's enlarge this. Okay, so we will try to blow this thing up. Okay, so now what I have done just now is I have only selected residue number 20 here. Okay, because it consists of several chain. Okay, so every individual chain, the residue number 20 is being projected. Okay, so if you want one particular chain only, so we will need to have a keywords of chain. Chain, let's say, what do you have? We have chain K, is it? Yes, we have chain K just now. Okay, is it? Oh, sorry, this is not. I think we have to. Okay, so if you type in only chain K, so it will only show us whatever which is belongs to chain K. Okay, so this is how we actually do a selection of items. Okay, so now if you want to label one particular residue, okay, what you need to do here is you need to go to mouse, label. We are labeling one particular items. So what you need to do here is go to your display windows, use your mouse, click on one specific point of atom that you would like the software to label. So they will label it for you. Okay, so this is ASN 507, number 572. And this is the nitrogen delta number 2. Okay, so if you want some others, then probably you have to click into another atoms. This is a valine number 584 oxygen atoms. So I repeat, how you label, you can go to mouse, label, and atoms. If you want to label a bond, okay, bond means that we are measuring between one point to another point to a bond. Okay, so what you need to do is go to mouse, label, click on to bond. What you need to do is you need to click into 2 points. Okay, so in order for the system, for the program to actually capture 2 points for them to measure the bond. Okay, so I'm going to show you how I do it. Okay, so I'm clicking 2 points, but probably you will ask, where's the bond? I couldn't see. The reason is because the bond is by default, the bond is being labeled, is using white, okay, as the label. So since we have changed the background into white, so you couldn't see the bond. So now what I need to do here is to change the color. We have to go to graphic, colors, okay. Label, we have to go to label, bond. Okay, you look at bond is being labeled as white. So let me change it to green. Okay, so now you can see it's being labeled with a bond, and the bond length, the distance between these 2 points, okay, is actually 8.59 angstrom. Okay, so same goes to mouse, same goes to if you want to label angles. Okay, so what you need to do is you need to click to 3 points, okay, in order to capture the angle. Okay, so I'm not going to go through that. What else I need to cover? Let me see. Okay, I think this
Excuse
one.
me.
Yes.
Sorry to interrupt.
Yeah.
Currently have a few questions, maybe need your help.
Okay.
So
Sure.
first is the the currying method in VMD can be based on the secondary structure. What is the reason that makes the protein has the secondary structure, and what is the difference between the primary structure with the secondary structure?
Oh, okay. I think there's several questions there, right? Okay, so let me address the questions one by one. Okay, what is the question? Okay.
Sorry, the currying method in VMD can be based on the secondary structure.
Yep, correct. It's based on structure.
So what is the reason that makes the proteins has the secondary structure?
Okay. In biology, in biology, the basic concept of biology, proteins. Okay, in nature, the proteins will have a primary structure which is consists of the sequence of amino acids residues. So in nature, this primary structure, the protein will fold into the secondary structure, and the bundle of secondary structure will be captured, and the protein will fold into the tertiary, and with tertiary structure, which is in a three-dimensional way. And if we have a several scaffold of a tertiary structure, it will be in a quaternary structure. Okay, that is the nature of the proteins. So what makes the protein fold into a secondary structure? I think this actually is the energy. I would say the nature of this protein that being in order for them to what you call to exist naturally. Okay, every single amino acid residue behave differently. They will have their characteristic of hydrophobic, hydrophilics. Those that which is hydrophobics, which is scared of waters, will be folded, okay, into inner part of the sequence, or I would say it will fold and hide inside. And those that having a hydrophilics properties will be hydrophilics means that they like water, means the amino acid residue will be actually happily exposing themselves into solvation conditions. So in nature, because of the nature of these amino acid residues, it will be folded naturally into a three-dimensional way, and it will be in secondary structure, which is alpha helix, as well as in a beta sheets or coil or loops, and a bundle of all these will form into a tertiary structure of the protein. So I'm not sure whether I address your questions. So
I think the next question is about the difference between the primary structure and the secondary structure. I think you already addressed the second question as well from the Hendrik.
Good. Okay. And I think I have another question is about VMD, right? Why the VMD is in secondary structure, is it?
Still have another question at the top. We go from the top first. Is there any way to convert the raw sequence data, one dimension, into structure, 3 dimension? If yes, how to do that?
Okay. Okay. Means that you only have a protein sequence, right? Okay. So this sequence, your question is how do we actually obtain or to predict a three-dimensional structure, protein structure from a primary sequence? Okay, this will go into another topics, which is protein structure predictions. Okay, so there are ways for us to actually predict a protein structure, three-dimensional protein structure, by using homology modelings as well as fold recognitions or ad initio prediction or de novo predictions. So for that is another one big workshops. Okay, if you are keen on that, okay, we will be having a workshops in BW Bioinformatics workshop Vestas, which will be running very soon. Okay, protein structure predictions, or we call what you call protein. I think we have a workshops on molecular mechanics, molecular dynamics.
Molecular dynamics and mechanism.
Yes, and protein structure predictions also will be included in one or part of the workshops. Okay, so that you will need to attend. If you are keen, we will have the entire workshop covering how you can actually produce a three-dimensional predicted model based on a primary sequence of your proteins. Okay. Yeah.
I think, yeah, Hendrik said that it seemed promising, and thanks for answering. And then another question is from Mohammed.
Okay.
Sorry. Why we use different drawing methods to represent our data?
Okay. I would say, okay, different drawing methods will tell us different stories. For example, as you can see from the demonstrations, I have actually used a serve, okay, and also a secondary structure cartoons, new cartoons, okay, to represent that. Okay, so different representations will actually tell us different stories. If you are if I am only producing using the same structure, only using a self representations, okay? How much story you can tell from a self representations? Self means that the surface of the proteins. It only covers the entire surface of the proteins, okay? Nothing much you can tell. Only with using the self, you can only tell cavity, whether there's a cavity or how big is the cavity, okay? But if you add up another representation, which is a secondary structure, new cartoons representations, you will probably see, oh, this part of the protein is actually folded into a secondary structure, a bundle of helix coil. And this bundle of helix coil, can it act as a channel that pass things through? Or can this beta sheets having a contribution in terms of the structural stabilities? Okay, all this can be postulated if we represent the data in a different way. Okay, so that is the reason why it's being presented in different representations. Okay, I hope this answer your questions, Mohammed, if I get your name correctly.
Yeah, I think so.
Okay, do we have any other questions?
Currently, no. I think you can continue.
Okay, so I think I will move on to the last part of our practical, because I don't want to spend too much time on VMD. I think my handout, okay, is comprehensive enough for you to follow. The handout that we are producing here is actually, we are trying very hard to make the handout to be idiot proof lah, okay? So we have all these screenshots, okay, for you to actually follow step by steps, okay? So that even with a non bio background participants, they can actually follow and go through it, okay? And by only following the instructions there, okay? Probably you don't understand the theory behind, but that will need Bio 101, Biology 101, but somehow if you go through the handout that I'm distributing, you should be able to follow the guide. Okay, so the last part that I'm going to show you is how you can actually render the images. Okay, so now going back to our graphic representations, okay, we will go into representations here. Okay, let me change my representation back to surf, put it into transparent, and this line, I will need to go back to our protein, okay? And line doesn't actually give us a lot of story, so I would like to change it back to our new cartoons, okay? And okay, probably you will see that there are some labeling that I have done, okay? So in order for us to give a clean images that we will be rendering in a while. So I'm going to show you how I can actually turn off the labeling. Okay, so now let's go back to our main. You will go into where already? Let me see. Label, graphic, labels. Okay, so when you pop out a label panel here, you will see these are the items that we have clicked and the tools have helped us to label just now. Okay, if you have, I can actually turn off, okay, all this. Okay, so these are the, okay, sorry. These are the residue that I have labeled. Okay, so now I'm going to hide it, or I can actually delete the label. Delete the label doesn't mean that we delete the item, so don't worry. You can delete, or if you still want to maintain it, retain it, then you can actually hide it. Okay, there are some more. It's still there, so I actually hide it. One more. Okay, where's the other? I think the other I labeled. Okay, so I'm going to, oh, that's the bond that I have actually labeled just now, right? So we will go into the bonds. Okay, also hide this bond. Okay, so far so good. Okay, so this, okay, so our image is clean now. So we are going to close this, and I'm going to put it into center, turn on my quick serve, and also my cartoons, new cartoons. I would like it to have a coloring method which is in secondary structure. Okay, so rotate it. I don't want it to be so big. I want it to be like this. Okay, so now we are all good to render the image. So where you need to go is you need to go to File, Render. Okay, so what you need to do here is you can actually choose your tachyon. There are different rendering methods. Okay, you can play around of each of them. Okay, try to exploit yourself which one give you the kind of image that you want. This is a rendering image, so you can go to tachyon and then select where you want it to. Here, file names. Okay, so you can go into where I want it to be. Just go to my probably desktop, or go to my documents. So let us try a BWF tachyon. Is it tachyon? Yeah. So you can choose TGA or bitmap. Okay, for those who have using a Windows, you can always choose a BMP, like what we have been sharing in our. Let me show you my handout. What I do for rendering. Okay, I didn't cover this. It's because we are running out of time. Then probably you can go on with this by yourself. Okay, I've shown you the bond labeling. Okay, so now we are going to go into the last part, which is the basic rendering. Okay, so you can play around with that. Okay, I think I have covered some of this, not all. Then this is the rendering. Okay, so you can go into this. I think this one is from Windows. Okay, means that the Windows system. You can go into tachyon, choose a tachyon. You can go into this, and then for the rendering commands, you can actually choose a bitmap. Okay, and by following this instruction, you should be able to render if you are using a Windows program. Okay, so try it out. If you have questions, you can post this to our moderators. Okay, I will try my best to figure out how to help you. And for a Mac program, okay, you can just choose. Let me try whether I can. And because I used to have problems on this. Okay, so let me do a BWF testing image. Okay, test image. Okay, so I want it to be BMP as well, safe. So here what I want is I want it to be BMP as well, but I'm not sure whether I can I can do that, because I used to have this problem that I can't. Okay, so you can start rendering. Okay, it will take a while. So let's go back to our script, interactive script VMD console. So it will tell us that BMP, I should have, okay. So let's try the other methods. You see, here will actually tell you in this windows, it will actually tell you whether the file actually successfully done or not. If you have a Windows, you will see that they will tell you rendering complete and the file, where is the file? Okay, so here you look at my error, there will be a what you call does not exist. So let me try again with some other snapshots. Okay, just try a simple snapshots. Okay, so now I will have a simple snapshots. So just start rendering. Okay, so you will have a simple snapshots like this in if you are using a Mac system. Okay, so this is how you have your image now. Okay, so you can actually integrate this image into your Word document, into your slides. Okay, so this is how rendering is done. Okay, so moderator, do we have any questions?
No, for now.
Okay, so if we don't have any questions now, it's 11:04. Okay, I'm going to wrap up the VMD parts because now it's 11. We have about 1 hour to go for the next two tools that I'm introducing. We have Pymol and also we have Ligplot. So I'm going to stop for 5 minutes break or 10 minutes. I will give a 10 minutes break for everyone for these sessions. So let's come back at 11:15. Okay. And if you have any questions, do post in the chat box and I will try my best to answer it. And we will resume back our session for Pymol in 11:15. Okay. So that's all for VMD.
Okay, now it's already 11:15, so I think Dr. Sibing, you can continue for the next session.
Okay, thank you, Choi. Is everyone back? Hey, wait, I think I haven't turned on my video. Okay.
Sorry to interrupt.
Yes.
Before starting for the Plymo, there's a question during the break.
Okay.
So
sure.
that is from Winfred, if I'm not mispronounced. So thanks for the interactive session, has been very informative. I'm just curious, does VMD support scripting in such a way I can automate the task in any language, either Python, Bash, C++, or others?
I am not so sure. Okay, the one that I can confirm is you are allowed to integrate with scripting, but the scripting might not be C++. Okay, so you will need to find out the scriptings from, let me see the scriptings from the what you call this website, where they will have some example on how you should actually put in your scripts, and how you can integrate it into the plugins, and then so that to execute. Okay, so for sure you can, but I'm not sure whether it can actually support the one that you have mentioned, which is C++. Okay, I hope this answers your questions. Any more questions, Tom?
No more.
Okay.
You may continue the Plymo. Thank you.
Okay. So, I have 43 participants now. Okay. Okay. So let's move on to our next visualizer. Okay, this is Pymol. This is what I have recently acquired in Pymol, how to use it, how to play around with it, and then I am actually fascinated by the kind of images that are produced by Pymol. I know Pymol exists for quite a very, very long time, and even before maybe at VMD, but somehow I didn't get a chance to actually use it during my, I would say, during my research, during my PhD time. So this Pymol is where I recently picked it up, and to be frank, I start to like it. Okay, so there is something which VMD cannot do, I would say, and the user-friendly level of this Pymol is higher than, I would say, higher than VMD. Okay, and I guess for those who have some knowledge in bioinformatics, I think a lot of the people out there actually use Pymol to generate some images, or even do some other analysis using Pymol. So yeah, so we will use Pymol here. Okay, a very basic usage of Pymol, I would say, and the same similar way that I prepared my tutorial or hands-on material is the am I sharing my screen? Wait.
Yes, you are sharing your screen. You are sharing your screen just now, but your
okay.
background sound is quite loud.
Oh, okay. But I didn't move my laptop at all. Let me see. I'm not. Okay, is this better?
No, the background sound is still there.
Wait. Let me do something. Wait. Okay, is this better?
Yeah, I think better.
Good. Thanks for alerting me on this issue. Okay, I hope everyone can hear me clearly.
Yeah.
Okay, so let's go back to my screen sharing here. I'm sharing my screen, but you are not. Are you seeing my Pymol material?
Yes.
Oh, you are. Okay, good. Okay. So, okay, back to what we have left just now. Yeah, the installations. Okay, so Pymol have recently launched their 2.4 versions. So, as this Pymol is freely available and it is also running across different platforms. So, I don't think if you manage to install VMD, I don't see any issue installing Pymol. Okay, it will be quite straightforward, I guess. Okay, so let's continue back in launching the Pymol. Okay, for me, I actually put my Pymol in the applications. So for those who are in your Windows, I guess by just double clicking your Pymol icons that on your desktop or either in your program file, you should be able to launch your Pymol. And yeah, I would like to say something on this. Pymol is freely available for educational purpose. Means that if I'm not wrong, you need to have a education, not a commercial domain in your email address when you register yourself. If you don't have that, probably you will have only the trial versions, which last you probably for 30 days, I guess, or 10 days. So for me, because I am affiliated with Perdana University, so I am eligible for education use Pymol. Okay, so this is how my Pymol looks like. Let me arrange this thing nicely so that my Pymol is here. This thing is sitting right at the side. Okay. So but this one is not my Pymol. This document is okay. Oh, I think I am having my Pymol in another Windows. Yeah, okay. Sorry, let me do it one more time again. I need to turn off my Pymol because my Pymol is in another desktop. Okay, so I need to close this as well. So my Pymol is here, and then I need to go to my program, applications, go to Pymol. Okay, so I will have my Pymol, and let me put my Pymol side by side on, and this thing side by side. Okay, so I will have my Pymol launch as what you've seen on my screens. So basically, Pymol have two parts where we have an external GUI here and a viewer here. And if you go through the tutorial, I have explanations on all the tabs and buttons that you have in this GUI interface, Pymol GUI, graphic user interface, where you have a command lines where you can type in your commands, and you have a movie control, which is right at the bottom here. Mouse matrix, okay, showing you whether you are doing translations and moving along the XYZ axis, okay, and you have object control panels, which is right here, okay. So now what you need to do here is in your object control panels, if you can see here, we have A stand for actions, S stand for show, H stand for height, and L stand for label, C stand for colors. Okay, so whatever actions you want to do here, like zoom, centralize your molecules, put it back into the origin positions, so you can actually play around with the action buttons. Throwing away the waters, removing waters, okay, delete some of the selection that you have highlighted, okay. So all this happens by using actions buttons, okay. So the next one will be show, okay. This is something very similar with what we have in VMD, okay, where the graphic panel, if you still remember, I will be using, I'm using VMD as an example is because I start off using VMD before I use Pymol. So that's why a lot of comparison, I tend to say, if you use VMD is in the graphic panels, okay? So this is similar with what VMD have in graphics. This is similar with what VMD have in display panel, for example, okay? So, the comparison I have done is comparing between Pymol and also VMDs. Okay, so now let's say show, you have all these like different representations, wires, line, okay, likerized, ribbons, cartoons, and all this. Okay, and if you move to H, which is stand for height, so which means that you want to hide particular representations, so you will be playing around with height buttons. Then label, okay, label one specific residues, chain, okay, and atom names, and one letter represent all this, okay. So you can play around with it, okay, no harm exploring. Your laptop won't explode, okay. Your program, worst case is it get corrupted, okay. If it's corrupted, what you need to do is just reinstall the program, so it won't nothing will die, or so you can just play around with it, explore it, okay, try one by one, okay. That is how I learned, okay. So I'm sharing my learning experience with you as well in this workshop, is where be brave to explore, okay. The computer won't explode, okay. Worst case is just the program corrupt, you need to reinstall, that's all, okay. Nothing, you got nothing much to lose, okay. Just you need to take the initiative, the motivation to explore it one by one, then that is how you actually learn, okay. So we move on to label, this is label, and the last is the color, okay. So this is basically the control panel that we will be using. I have a very detailed explanation also in my handout, so you can actually go through it. So then we move on to the similar way that we are going to load our structure into our Pymol. So what we will do here is there's actually two ways to load the molecule into Pymol. Okay, one is to fetch the molecule directly. So what Pymol will do for us is where they will actually go to the website to fetch the molecule into our visualizer, Pymol visualizer. Or you can also browse the file like what we did in VMD, where we go to what you call file, and then after that browse molecule, and then go to the directory to actually load in the molecule. Okay, that is what VMD will do for us. But Pymol is slightly smarter. Okay, they have a scripts, okay, in Pymol, where what you need to do here is by typing fetch 1RUZ, okay, Pymol, let me close this. Pymol actually have a script to actually go into rcsb.org. I can't remember RCSI, sorry. rcsb.org/1RUZ. They will actually go into protein data bank directly and to download the PDB file for us automatically. So fetch command, this is a plugin that written in Python scripts that fetching the structure file from protein data bank directly. Okay, you can also use this way. Go to File, Open the PDB file where you just downloaded just now. Okay, one RUZ, and then open. The file will pop up as well. By using fetch plugin, it can also fetch the structure directly into our visualizer. So I'm going to do something different here as compared to VMD, where we are going to use fetch here. I'm going to show you fetch. If you want to try the other way, you can try it out on your own. So we are going to fetch our one RUZ. Okay. Are we? Okay. Okay, so right away we will have our one RUZ.pdb being loaded into our visualizer. Okay, so you will have, if you have your Pymol ready with you now, you will see that one RUZ is being loaded. Okay, so now we are going to play around with the representations as what we did earlier on. Okay, so what should I do here? Okay, so I'm going to show you something where VMD has it, but I didn't show. Okay, but now I'm going to show what Pymol also can do with simpler way. Okay, if you can see a button here at the bottom, there is an S here. So by clicking here, the S, this is actually a sequence panel. So by clicking here, you will see the sequence of this structure, one RUZ. Okay, it actually tells us that from here, okay, how many residues that it make up of. If I actually blow this up, okay, let me see. Okay, so from here you can actually see total, there's how many chain of it. Okay, I remember just now I saw there's what K, L, M. Okay, this is I chain, and we have chain A, which is H chain. Okay, some more. Let me go through one by one slowly. Okay, H chain from residue number 1 until residue, if you scroll it down, residue 321 plus something lah. Okay, so this is one chain. We have chain B. Okay, the residue number starting from 501 onwards until 656. We have chain J. Okay, this is from also residue 6 until 321 plus. Okay, D, and let's go on and on and on. And all these 0, 0, 0, 0, 0, all this, guess what? This is a water molecule. Okay, this is a water molecule. Okay, so from here, immediately you will know the sequence of one RUZ hemagglutinates. Okay, so what else we can do? Let me go through the hands out. Okay, so we are going to play, explore something on the representations where okay, there's different representations that you can play around. So we can go into this part. Okay, the what you call, this is the control panel, object control panels, where you can play around with representations. Okay, you do see that there is a selection here because I actually mouse over some of the residue. That's why it's being selected. So now I am going to remove these selections because I don't want this to confuse everyone here. So I'm going to delete these selections. So now, if you are the one that loaded the one RUZ into the visualizer, you should be seeing only one RUZ. Okay, so now I am going to show you show cartoons. Okay, this is what I like. This is already in cartoon, sorry. And I am going to show also the spheres, probably. Surface, sorry. Okay, so this is surface together with our spheres, and also hiding behind it's the secondary structure thing. So now I'm going to hide my spheres just that I have just created. So I'm going to hide it. So now what you can see here is you can only see the surface and also the secondary structure which is hiding behind. Okay, so now what you can do is you can actually play around by choosing if you want your structure to become lycorized, then you can show lycorized. But before that, you need to actually hide the surface. Okay, then you can see your structure being displayed into lycorized. And you also need to hide your cartoons. Then your structure will be only showing lycorized. So this is slightly different than VMD. For this part, personally, I think VMD is smarter lah. Okay, Pymol, this is a bit like a bit complicated. Every time you need to actually hide the representation one by one. Okay, so you can change the color. Okay, you can change the color into let's say if you want it to be red, the entire thing will be changed into red. They have a lot of presets color that allow you to play around. You can color it by chain. Okay, so different chain will have different colors. So you can color it by secondary structure. Okay, so secondary structure, they have several options for you to choose. You can have a helix in science, okay, sheets in magenta, loops in, I don't know what you call pink, okay. So there's different options for you to play around. So in terms of coloring methods, I would say Pymol have a better options for you to play around. Okay, so you can have different colors. So show in cartoons, okay, but you need to hide your lycorize first, then you can present it in, okay. So and okay, so this is the way to actually maneuver the object control panel. Okay, so from here, I think you can have a brief idea how we can actually play around with the control panels. So there's a best thing for Pymol is where if you go into these actions, okay, there's a lot of presets, okay, presets for you to play around, okay, means that they predefine how you should actually represent a proteins, okay. If you go into actions presets, you will find a lot of way, like for example, simple means that it looks like this. I'm sure maybe you have seen this kind of representation before. If you play around, if you go to presets, publications with solvents, then you can see it's being represent in different colors together with the solvents. Okay, this is very nice. And if you go into presets like publications, ligand site, okay, then if you want it to be have a solid surface, then they will actually show all the ligand binding sites with a predefined kind of representations. So you're going to it's not necessary for you to actually you don't even need to be creative, okay. Pymol does it for you, okay. So they actually predefine the binding site according to the structure, okay. And they I should rephrase my statement. They don't predefine the binding site, okay. The structure define it, the protein define where is the binding site, but Pymol decide how the binding site should be represented so that we can understand how the binding site looks like. Okay, so by going into actions presets, action presets. Okay, so there's a lot of options for you to play around with. Okay, so if you want to look only with protein surface, okay, so let's just hide our glycolides, I think. Okay, so this is how the surface presets B factor. Okay, this is about B factors. So B factors is parameters that we actually look at how flexible or the amplitude of the vibrations when they do crystallizations on one particular atoms. Okay, so when you look at this image, you will know that this is representing the B factors value. Means that this part is very flexible, where this part is very rigid. Okay, so this is predefined by Pymol as well. Okay, it's not necessary for you to actually crack your head to look at, to figure out how to actually represent a B factor value. So Pymol does it for you. Okay, so what else? Well, I guess the other representation you will need to exploit yourself. Okay, there's quite a numbers of presets that you can actually play around with. Okay, then yeah, we move on. Okay, so just now I actually show you about the sequence. So for sequence representations, you can actually go in. We have already clicked on the S, okay, which is the sequence panel that are being displayed out. Okay, so there is also some other representation where you can actually go into display sequence mode. Okay, now we are displaying the residue codes. So probably most of you might not remember what is the single letter representations of each and single amino acid residue, as there's 20 of them, right? So maybe you will prefer a chain a residue names. So once you click on residue names, it actually change into a 3-letter representation of each and single amino acid residue. So this will be easier for us to recognize, I will say. And what else? Okay, so there are several different chain identifier. If you click on it, you will know that which of the residue actually belongs to which from which residue to which residue actually belongs to which chain. And also atom name. Oh, this will be very long, where every single amino acid residue, every single atom which belongs to one particular amino acid residue will be presented in the sequence panel. Okay, so isoleucine, okay, you will see there is nitrogen, carbon alpha, carbon, okay, oxygen, carbon beta, all this being listed, every single atom. Okay, what else we have? Okay, I think that is basically, yeah. So now I'm going to show you how you can actually run through the structure analysis, okay, where you can do selections. Let's go back to our display sequence. Let's go to a sequence name, okay. Let's say, for example, you want secondary structure representations on a specific region of this protein. So what you need to do is, this is pretty simple, I would say. What you need to do is just bringing your mouse over. For example, you wanted to see selections of interest residue from position 503 to 511, for example. This is what in your tutorial is stated. Okay, so we can actually scroll to 501, okay, to 511. So let's highlight this particular residue. Okay, what you need to do is just go to this part 501 to 511. Okay, bring your mouse over and highlight this particular residue. Okay, so once you have highlighted this region, okay, you will see that one additional selections is being added in the control panel. Okay, this is the selections that you have just selected by using your mouse over, sliding through the sequence panel. Okay, so now this is the one that being selected. So, but this, you couldn't see it here. It's all small little red dots. So we would like to change this region into other different representations or other colors. So what we will do here is, if you have went through my tutorials or the hands-on material, you will see that we are actually using some command, okay, some scripts command that doing the color changes, okay, color the different atoms and all this. So I'm going to introduce that for you as well, because that is the strong thing. I mean, the strength of Pymol, that you can use a simple command, okay, and you will feel that, oh wow, I can actually use command line to call out some functions instead of clicking the buttons. Okay, so I think I would like all of you to try on that. So let's try something like color. Okay, we can actually color. Go into this command input area. You can type in your color. Okay, color blue. Okay, into before we do that, okay, let's give our selection a name. Okay, so I'm going to go into selections, rename our selections. Okay, so what we want our name to be is, okay, let's give the selection the number of the name of conserve. Okay, conserve rack. Okay, so we will have our selection with conserve rack. Okay, so once we have that, then what you need to do is, okay, go back to our command input area, type in color blue. Okay, and where you want it to color, conserved. Okay, conserve reg. Okay, so you will have conserve reg. Okay, sorry. Okay, what name I'm giving? Okay, sorry, I have a typo here. Conserve. Okay, once you have that, you will see that all this region is being colored into a blue color. Okay, so this region is being colored. Okay, this is as easy as. So this the script that used to color it become blue is actually the plugin of color command. Okay, the word that you are typing here, color, okay, is an instruction that telling Pymol, okay, to color that part, to color blue into conserve red, which is the selection that you have just selected just now. Okay, and behind the color instruction is actually a bunch of scripts that telling the Pymol to color it. Okay, so nothing fancy. Okay, you are telling the software to do this work for you. So you are the owner of the instructions. Okay, so this is what on color. So you can also change the surface, okay, where I have already shown you just now. So what other things that you need to do? Okay, let's go to labeling. Okay, so how you can actually label this part. Okay, so you can, okay, use the command as well. So now let's go to the command that we are going to use is label. Okay. Resi. Okay. Residue. Resi stand for residue 503 to 511. Okay. And name, just follow the syntax that given in the manual, then you will get the labeled. Okay, so what I have here is this part where you need to type carefully because the program is following the syntax. So if the syntax is wrong, so you couldn't work. So the best is where you copy and paste. Okay, you can put the thing side by side and then you copy and paste it. Yeah, why my one is not labeling? Because the syntax is not correct. Huh? Okay, let me see label 50. So for me, most of the time, I will copy and paste this thing. Okay, I think I miss S here. Yeah, correct. Okay, so from there, you'll see all the label is being labeled. Okay, so now let's say if it's too messy, I guess, so I want to hide this thing, hide the label. Let me see, can I hide my label? Hide label. Okay, so the label is too messy, so I just go back to my main structure, okay, and hide the label. So if you don't want, you can actually show the label again. So go to show. You have show label. Oh, you need to label again. Okay, so you can actually label again. Okay, so this is how labeling is being done. And in my manual, I have also share some of the way how you can actually set your labeling. You can change the font size of the label, okay, by going into the setting, okay, and labeled, okay, setting label size. So now currently by default is actually a 14. So you can actually make it smaller, then it becomes small. Okay, so you can play around with the labeling. And you can also change the colors of your label. Okay, so I guess you can follow my tutorial to play around with that. And yeah, so I think I want to skip all those and move on to the last part where we will have the rendering of the images. Okay, so how you can actually render the image. So don't do print screen, okay? Print screen will not give you a high quality of the structure, okay? The visualizer will have better way to do all this, okay? So now what we will have here is we can hide the label now because it's a bit, I will clear the label. And okay, so now to save the images, okay, what I will usually do is I usually go to draw. Okay, so from here, okay, immediately you can actually play around with the DPI. Okay, this is how you can actually set. For example, publication, most of the publication actually needed 300 dpi at least. Okay, some even go into 700 dpi. So you can actually directly change your DPI here. Okay, and you can also make your structure to become a transparent background. Okay, so just need to, what you need to do is just check on the transparent background and then just click draw. Okay, so where is my file? Why I couldn't see? the file is being, okay. So you will have an image like this. Let me see. Okay, so you have a image generated out. Okay, so you can go to this file. Okay, what you call, you can do save image as, okay, so you can do PNG. So it will have a save image. Okay, so what you need to do here is just type in your file. Okay, BWF underscore what Pymol test. Okay, so we'll have our, okay, so to check our file, whether our file is there. Our file is at our document, right? BW. So it's there. So this is how our file will look like. Okay, so that is how easy we can get our image being rendered. Okay, so there are also videos that you can actually play around with. Okay, where maybe I can share, I can show. You can go, let's say for example, you would like to save a video. Let me see whether I can do it or not. Where already? Oh, sorry, this is not my this is not my Pymol Windows. Okay, so you can actually save movie as, okay, you can do MTG. Okay, so what you want them to do for you is you will have a movie, but before that I think where I can get, okay, you can do this. You can go to this rock. So your structure will be moving. Okay, and orange. So let me see whether I can produce a movie. Okay. Reset. Okay. So we'll go back to our file. Save movie as, you can have a MPG, MPEG 4, and then save movie as. Then we'll have here, let's have a Pymol, no, so BWF Pymol movie. Okay, and let's save. Oh, yeah, I have one earlier on, so I'm going to replace it. One frame, oh, because I'm having one frame. Okay, so now, okay. Okay, so my movie has only one frame. Okay, so now let's see. Okay, so I will have a movie. So you can put, once the movie is generated out, you can actually insert this movie into your slides. Okay, that is how the movie is being generated. Okay, so I guess for Pymol, we have to stop here. It's because it's already 12. And we still have one more to go. So I guess I need another about maybe 20 minutes to run that. So I guess we will have another break, but this time we are going to have only 5 minutes for Pymol. So sorry, we are going to have 5 minutes break for after we have wrapped this Pymol ups, and we will resume back in 12:04. Okay, 12:04 for our Ligplot. So for those who have any questions, please drop it into the chat message, and then I will answer it when we resume back before we kickstart our Ligplot. Okay. Okay. Thank you.
Hi, Dr. Siving.
Yes.
Now
Let's
already
bring back that.
12:04.
Yes. Yeah. Let me.
There is some question from YouTube and YouTube channel.
Yes.
So first question is what is the size of the image is usually preferred as standard?
Size preferred as standard, you mean in terms of publication, is it? I assume yes. Okay. So there's not really a limit in terms of the image size, but the resolution, yes, means that the DPI. Okay. So usually the publications requires at least 300 DPI. Okay, means that the fineness of the images generated out. So there's a trick that you can actually check on this is where when you enlarge it into 200% or 300%, your image should not be pixelated, I would say. Okay, then this is considered acceptable. That's a general way to check whether your image is acceptable or not lah. Okay, so what you can do is try to enlarge your image. First of all, you need to ensure that your image have at least 300 dpi or even higher. The higher the better. And the resolution, I'm looking into the resolution of the image. And after that, how you should check whether it's acceptable or not, then you can actually enlarge your image into 200% or 300%, and then to see whether there's any pixelations happens in your image when you blow it up into to 200 or 300%. If there's pixelations, means that usually the editorial board will reject that kind of images. Okay, so to prevent that from happening before they review your journal, your manuscript, you should ensure that your images should be at least high resolution, no pixelations happens. Okay, so in terms of size, I don't think there's limits, but usual common practice is where we try to make our image within a A4 paper size, because that is the usual way of us reading a journal, right? We'll print it out into an A4 size and then we read out. So we have to ensure that it's readable with under 100% enlargement. Not to say enlargement, the original size, which is 100%. And this is within the size of an A4 size. Okay, that is what I will usually do. Okay.
So the second question is, instead of aligning the label behind the protein structure, can we done beside the protein structure?
Yes, you can. You can position your label to a designated place that you want. Any place that you want, you can position it. In VMD, I have experience in doing it. But Pymol, to be frank, I don't have any experience yet, okay? But I'm sure it's doable in Pymol, okay? So we can actually position the label, but there's also one experience that I would like to share with all of you is most of the time I don't use the visualizer to label the item, the thing for us, for me in my image. I will use either Photoshops or PowerPoint, okay, to actually help me to do the labeling, okay. So because that will give me freedom where I want to label it instead of, okay, if you use the visualizer to help you to label, if you turn the structure around, the label sometimes will be blocked by some other chain or some region of the structure. So I don't want that to happen. So usually I will render the image and then label using some other tools like Photoshops or Illustrator or even Paint. Okay, the Paint in our Windows, I use Paint to label it. Okay. Next questions. Do I have next questions?
Yeah. previously, do you have do somehow the movie on slide for the Plymo? If yes, so I think Winfred, if I not mispronounce, so maybe you can just look back for our YouTube link for the, and then you can see how to have the Plymo impact file, the movie on slide. Since just now Dr. Sibing already have did it.
Yeah.
Thank you, everyone. Yeah, that's all from the for the question.
Okay, good. Thanks for all the questions. Indeed, I actually like this kind of interaction, although it's not a real time, there's questions, but at least I don't have a silence group that don't ask questions. Good. Okay, so we are running out of time. So let's us move on to, I think someone is entering. So let me admit, okay, admit. Okay, so let us move on to the last part, which is Ligplot. Okay, so Ligplot Plus, as what I have introduced earlier on in my briefing or lecture earlier on. This is tools that help us to actually do interactions analysis, where this tool have predefined how a hydrogen bond will look like, under what condition we'll have hydrogen bond, under what condition we'll have a hydrophobic interactions, under what condition we'll have a bye-bye interactions. So all these things is actually predefined in the tools. Okay, so what we need to do here is where we will just need to upload a coordination file, okay, into the tools. Coordination files means that a PDB structure. What PDB structure consists is only a coordinations of each and single atoms. You remember I earlier on this morning, I just shared with you a whole set of numerical numbers with maybe residue names, okay? So that is actually a coordination file of each and single atoms, okay? So what the program actually do for us is to translate all this coordination into something where you can visualize. So VMD have done that, Pymol have done that, same goes to our Ligplot, okay? Ligplot plus or Ligplot is actually measuring all the coordinations and using the predefined rules that in their program, they tell us, oh, between this atom and that atom, this is a hydrogen bond. Within this distance or this bond angle, oh, that is being defined as a hydrogen bond. And when I see this residue, this is a hydrophobic residue. So I give them a hydrophobic representation. So that is how a Ligplot generate a 2D interacting diagram for us. Okay, so let's move on to our tutorial. I have my, wait, let me share the screen with you all. I think I accidentally closed my Ligplot tutorial. Let me see. Okay, so okay, so this is a Ligplot tutorial. I have done for everyone. Oh, I guess I need to share my. Well, I need to share my desktop. Sorry. Okay, so this is my desktop. So I'm putting, okay. So this is the Ligplot. Okay. I have received, no, I should say the organizing committee have received quite a number of emails saying that you are having difficulties in installing the Ligplot. Okay. So Ligplot needs you to register. Okay. Once registered, I guess you need to log in with the password. Then only you are allowed. Immediately they will prompt you. They will download the program automatically. Okay, so and once you have downloaded, that is actually a zip file. So okay, so this zip file, what you need to do is you need to extract this out. Okay, and put it into wherever is wherever you like, okay, the directory that, the path or directory that you like. And then after that, you just need to go into that directory and double click on the jar icons, and then you should be able to execute that Ligplot. Okay, so for Linux and Mac user, this will be slightly complicated because there's no way for you to double click the thing. You need to issue command in order to execute the to launch the program. So so I will be showing you how this will be done. Okay, so installation, I am assuming everyone managed to install it in Mac or Linux. Okay, so then so to execute it, okay, so this is the command to not execute, to launch the program. This is the command that in Linux or Mac is used. In Windows will be easier, just double click that thing. Okay, so I am going to execute it using my terminal. So now how I'm going to launch a program is by issuing a command like this. Where's my liblock? Okay, too heavy. Okay, so once the program is being launched, you will see like this. Okay, and I actually highlight the path directory. Okay, okay, so this part, okay, I guess if problem happens when you try to launch the program, this will be the part that everyone have to take notes that I would like to highlight here in this tutorial. So to launch your Ligplot, okay, a temporary directory is needed. So you will need to tell, you need to first of all create a directory giving a name TMP, okay, and you need to direct the program into that TMP directory. Give the path of where the TMP directory is located. For example, I put my TMP in user cbing document /temp. So for Windows, probably yours one might be in D, okay, and you create a TMP empty folder. Just create an empty folder, and you need to tell the program where this empty TMP folder is located by paste the path into this part, which is program path. Okay, you go to the edit, program path. So you'll be putting this thing in. Okay, and the second important part that you need to edit is where you will see that I actually integrate the Pymol into the Ligplot. Okay, so you don't need to, if you it's not necessary for you to actually integrate this, okay, but with the integration you will be able to see the three-dimensional kind of interaction instead of a 2D diagram where you have seen in my slides earlier on, where the one that with a spiky thing, okay, with a dotted line and then with some residues there. So that is a 2D diagram where Ligplot will generate that thing for you. But if you integrate it with Pymol, you will be able to see a 3D diagram. Okay, so I don't want to further extend this. Okay, so we will just cancel this one. Okay, you set the path accordingly. So very easy for Pymol, for Ligplot. Okay, so what they need is only coordination file. So what you need to do is just go to open PDB file. Okay, you can browse your file. Okay, or you can just type in the PDB code. Okay, and Ligplot will actually fetch that file for you as well. Okay, so for me, I can just type. They will just fetch that file for you. Okay, once the file is being fetched, so you will see a pop-up windows. Okay, there's some lagging here. Okay, so you will see a Ligplot or /thinplot. Okay, windows being pop up. Okay, so with the coordination file, Ligplot will know which one is the ligands. Okay, ligands, which is the other structure or other molecule that which is bounded with the proteins. Okay, ligands. So from here, we'll just, all this is being identified as a ligand. So this is a demonstration. So I'm going to share with you only by selecting one specific ligands, okay? And I'm going to just pick the first one, and then to tell the program, this is the ligand that I want to see the interaction, this ligand with the protein, 1RUZ. Okay, so I'm going to run it. So they will generate an image like this. Okay, so I'll explain a little on what this diagram means. Okay, there's a legends that in the manual that I actually put in to explain what are all this about. The spiky or the eyelashes, I always like to call the eyelashes representations, okay, because it really looks like eyelashes. Okay, so this is stand for hydrophobic interactions, and this is our ligand actually. And we have one particular amino acid residues, okay, number 68, is having a hydrogen bond interactions with our ligands. Okay, so that is how simple this is being interpreted. Okay, hydrogen bond, hydrophobic interactions. Okay, but of course, you need to understand how they actually tell you, oh, this is a hydrogen bond interactions. Okay, this is a hydrophobic interaction. So you can actually go into here. There's some runtime parameters that actually explain how they actually give you a hydrogen bond parameters. Okay, from here, okay, I think this is quite self-explained. Okay, they are actually telling you, they are actually measuring as long as they see a distance 2.7 within 2.7 angstrom, okay, between a hydrogen bond acceptor, okay, then they will tell you that this will be taken into consideration as a hydrogen bond. And in terms of donor, hydrogen bond donor is 2.35 angstrom. And for non-bonded contacts is 2.9 angstrom and 3.9 angstrom. This is a default value that's set by HB+. Of course, you can change these parameters. Most of the time, I will use 3.5, okay? And of course, this have to be backed up with the reference, okay? The reasoning on why is it you are defining a hydrogen bond with a 3.5 distance away, okay? So we can play around with this, okay? Of course, you need to be rational also. The hydrogen bond won't form above 5 or above 3.5 angstrom. So you can't simply just put in a value that and then to tell people that with 5 angstrom that hydrogen bond will form. Okay, so the default value is here, and if you have a strong justifications to change this value with a solids reference, then you do welcome to change all these parameters. You are allowed. Okay, so then yeah, that is how simple the Ligplot is. Okay, all this labeling, the size of the labeling you can change. Okay, you can go to the size of parameter, everything. Okay, this is one, the size of one. Okay, so you can try to exploit, play around with number. Okay, the font size, and then all this to see whether that is the size that you prefer. Okay, then what else? Okay, you can play around with this. Okay, now is the time where you save the file. Okay, you can write it as a PostScript file. Okay, so just go to file, PostScript file, and then the location where you want your file to be saved in. Okay, just BWF underscore Ligplot test. Okay, so you will have a PostScript file generated out. Okay, so you open it with PostScript readers or Acrobat readers, you can read PostScript file, as simple as that. Okay, so for now, you can see the example I'm using is 1RUZ. Okay, so most of the time when we want to study interactions, maybe we have a result gotten from docking, Autodoc, okay, Vina, or etc. Okay, so you want to study the result from docking, the interaction of the dock result into the protein. So how you can actually read this file? Also very simple. What you need to do is copy your ligands result, I would say the dock result of your ligands, the coordination file, copy it, paste it into your protein file, and then put them all together as one single PDB file and load it into our file. Then immediately they will define which one is your ligand. You just tell them which ligand you want, and then they will give you the exact image like this. Okay, so I would like to show you also the integration in Pymol. Once you set the path, like what I have done here, runtime parameter, sorry, not runtime parameter, my program path. Okay, so I have set it. Okay, that is where I actually install my Pymol. My Pymol executable file is located. So I have set this. So now I can actually visualize my interactions using Pymol. Okay, so I can show you. Okay, so once that, let me see whether they, is it here? No. I think it will take some. It will load, but let's give them some time because I'm loading two Pymol. Okay, so you will see image. Is it? Oh yeah. Let me close this. Let's. Is it out? Cannot be. Where's my Pymol? Is it? Loading. Come. Wow. Here. It's out. We'll need some time. So Pymol is loaded. Then you can see the interactions being projected in a three-dimensional way. Okay, so it's very easy. From here, immediately you can actually render the image and then do whatever labeling or explanation you want in your discussion, result and discussions. So this is, once you integrate it, there's two programs that integrate with Ligplot. One is Rasmol, one is actually Pymol. The one that I am showing here is actually Pymol. So you can, all of you can try on that. Yeah. So with that, okay, I would like to wrap this up. Okay, but before that, let me unshare this, and I would like to go back to my slides. Okay, here. Wait, let me reshare my slides. Okay, share my slides. Okay, so I have done all the demonstrations. So let me do a summary and recap here. Okay, so you have seen the demonstration by using VMD and also Pymol to generate images and also, and also videos. Okay, so why my, okay, so this is a short video that I have generated using Pymol earlier on, and I put it into my slides. A very short, I should have generated longer, longer rotations and orientations videos. So if you can see, this is how it's being done. But the summary that I would like to do is all the software actually involve three main steps, where get the structure file, okay, play around with the representations, and if you want the image, you can do file rendering. Okay, all these three steps involved in three different tools. Although they are different, but the process is the same. So I hope that you will enjoy, and if you have questions, please, moderator, Ms. Chong, do we have any questions so far?
Yeah, now have one question from French Danny. I'm using Windows 10 64 bits. When I try to run ligands on Ligplot Plus, there's a warning box saying that unable to create log file in temporary directory C10.
Yeah. Yes.
So
okay, that is that is what I have highlighted earlier on, but I can actually repeat this part where in your manual, you need to actually set this. Okay, let me share my screen now. For the here again. Okay. Can you see my?
Yes, we can see your screen.
Okay, it's in the tutorial hands-on, right? Okay, so this is the part that you need to set. Okay. The error telling you unable to create that log, because when they launch the interactions, when they analyze, they need a directory for them to actually dump their temporary file. So in the program path, by default, if I'm not wrong, they are actually putting in a C directory, and then they give a directory name which is TMP. But most of all of you here, or even include me, I don't have that file, that directory being created. You need to create that folder by yourself. Just create an empty TMP with exact the same name of this TMP. Okay, this TMP, create that. Okay, and give the path into this temporary directory. You need to tell the program where to look for that folder. Okay, you need the path. Once you have done that, then your program should run gracefully. Okay, so I hope I answer your questions. Any more questions, Ms. Choi? I think it's
no, there's no any question anymore.
okay. So if there's no questions, you get my email from, I think probably you can drop, if you miss the PowerPoint slides, you can actually drop email to the organizing committee to get my contacts, my email. And you have any questions, do write to me, and I will try my best to help all of you here. And with that, I would like to thank all participants for staying on throughout these 3-hour sessions. Thank you very much. And I hope that my workshop actually benefit all of you. Not immediate, probably, maybe you have some general ideas on how this, what is the functions of all these visualizers, how it can actually generate image or generate visualize your biomolecules. So I hope this actually help in your work and you do learn something. And for those who are still interested, please explore as much as you can. And if you have problems, always welcome to write to me. Okay, so thank you everyone. Thank you organizing committees. Thank you participants. And with that, I would like to wrap it up this morning. Okay, bye. Thank you.
Okay, so for the participants, if let's say you all would like to have the slide, please send the email to our AP BioNextSec Gmail. I will type the Gmail in the chat so you all can request the slide from us after this event. And then if let's say you all want to have Dr. Siving's contacts or anything, or BWF workshop, then workshop details, then just let us know from the AP BioNextSec Gmail as well. So do you still have anything to say?
I'm all good. Okay. Yeah, I just like to one more time thank everyone for staying on, and then I really hope that what I'm sharing this morning benefits all of you there. Okay, so for those who are interested, do join us in BWF. We have a series of workshops, okay, with reasonable, affordable pricing. Okay, the workshop actually runs throughout 24 hours. Okay, we don't run 24 hours one shot lah. Okay, so we are running it session by sessions. Okay, depends on the arrangement within the between the instructor and also the participants. So one do feel free to actually browse through that 20+ workshop that we have, and if you are interested, drop us email or sign up. Okay, we will let you know when the course will commence and who will be the instructors. Okay, so with that, thank you very much.
After this workshop, we will have a break, lunch break, and then after the lunch break, we will have the keynote talk at 2 p.m. GMT +8. So please check your emails for the link, and then in our welcome email. So we need to wait for 5 minutes more. Yeah, now Dr. Asif, are you there?
Yes, I'm here. So I guess we are done with the workshop.
Yes.
So I just have a quick announcement. I will just quickly share my screen. Can somebody enable screen sharing? All right. First of all, I want to thank Dr. Choi Sibing for the wonderful session. I've got a lot of wonderful feedback. So thank you very much, and working tirelessly, the night before preparing for this. And yes, thumbs up to you. A big round of applause to you. And I hope the participants enjoyed the session. And if there were any shortcomings in terms of links and connections, we do apologize. There was a question in the other workshop, and I just wanted to, say something in relation to that. You might have questions, and if you want to kind of connect to us and get some help, not just from us, but from the community. Of course, there are many ways to get help. One of the ways that we have been pushing forward is a data science interest group, where we actually have people from around the world, and with all kinds of interests, of course, largely bioinformatics. And we do share a lot of events, news, job postings and all that. And occasionally people have research questions or questions, they post there, and they get people from the members helping out. So if you're interested, you may want to take a photo of this, or print screen, or go to the link right away and join the group if you're keen. But do observe the etiquettes of the group. No spamming and all that. But yeah, if you need help, this is one of the ways. Dr. Choi is a member. Dr. Kenneth is a member of the group, and Prof. Tan, who is a keynote speaker, is a member of the group. So we do have a lot of luminaries in the field who are members of the group, and we'll try our best to help. This is one of the ways, not the only way. So you may want to take advantage of this. So with that, I thank you, and you all deserve a good break, especially Dr. Choi. And we hope to see you at the keynote session later. Do come back. The links have been sent to you. If you didn't get, I think we're actually going to send another email very soon, just so that you all have the links, and then there's no confusion. All right. Thank you.
Sorry to interrupt. Just now I mentioned wrongly the timing for the keynote talks. So the keynote talks will be start from 1:30 p.m. GMT +8, not 2 p.m. So it's 1:30 p.m. GMT +8. Thank you.
Thank you. So yeah, you have about less than an hour. Do have a good rest, and see you soon later. Bye-bye. Thank you so much.